have to talk about trihybrid cross. Now this cross demonstrates the principle of inheritance proposed by Mendel, also applicable for multiple traits. It's not only for one pair of traits, two pairs of traits, but many pairs of traits. And the Mendelian principle is also applicable for the law of segregation, the law of independent assortment, for more than two pairs of characters. So we have, for example, here I mentioned about for three pairs of genes, three pairs of contrasting characters or three pairs of genes. So the Mendelian inheritance is also applicable with reference to law of segregation, law of independent assortment for three pairs of contrasting characters or three pairs of actually genes. Now, we have to go for this trihybrid cross. So what is trihybrid cross? It is a cross between two homozygous pure breeding varieties which are differing in three pairs of traits or three pairs of genes. For example, we are taking actually three characters of the traits. One, the height. Another one, just you see that one, color of the cotyledon. Another one, the shape of the seed. So as the color is concerned, if you're taking the height first, I'll take. There are two varieties, tall and then dwarf. And color, yellow and green. If you're taking the shape of the seed, there are two varieties. One is round, another one is wrinkled. Another one is wrinkled. So in trihybrid cross, we are taking three pairs of contrasting characters. How far these characters are transmitted and inherited? So we are taking three pairs of characters simultaneously. So it is nothing but the inheritance of three single processes. Three single processes. Now trihybrid cross, which involves a combination of two single processes. It is a combination of three single processes operating together simultaneously. So this is one single cross, another single cross, the third one, what we have the shape. Now, so it's a combination of three single processes in contrast to two single processes for dihybrid cross. So all these characters are operating together. So we have to study the inheritance of three pairs of contrasting characters together. Now, here we are taking the example, a cross between a homozygous. Now we have to take the parents, so parental generation. So we are taking three pairs of characters, one with reference to what we have, the height. This is tall. Then we have, there is the shape of the seeds, that is round. And the color of the cotyledons we have, that is yellow. All pure homozygous dominant. One for the height, another one for the shape, another one for, that is what we have, the color of the cotyledon. The gene for height is located in northern one in the fourth chromosome. The shape of the seed is found in the case of, we see that 1/7 one. The color of the cotyledon normally occurs in chromosome number 1. Now, this is tall, red, tall, round, and yellow. Tall, round, yellow. So all are homozygous in nature. So it's being crossed with a plant which is recessive for all the characters. It is recessive for all the characters. So this is what we have, dwarf, that is wrinkled with yellow with the green cotyledon, with the green cotyledon. This is the nature of the parental generation. This is the genotype and this is what we call the phenotype. When cause is affected between, that is pure homozygous individuals which are differing in 3 pairs of characters. We have received the F1 generation. We have received the F1 generation. So, based on the independent assortment, we have received actually a trihybrid. And the trihybrid is having a gene for round, tall, and also that is yellow. It's normally all these are all dominant character. In the F1 generation, we are getting what is called a trihybrid. So this is for what is called the tall character. The next one, this is for round. And the next one, just what we have, yellow. This is called trihybrid. The receptor lineage is called trihybrid. This is F1 generation. We have trihybrid differing in 3 pairs of characters or 3 pairs of genes. Now this is a trihybrid. Now in your syllabus given only what are the different types of gametes produced and what is the phenotypic ratio obtained in trihybrid cross. That's all. There is no checkerboard, nothing else. Now a dihybrid individual produces 4 types of gametes and the types of phenotypes in F2 is also equal to 4. So 4 types of gametes and equal to 4 types of individuals in F2 generation. Now here we have 3 pairs of genes differing, 3 pairs of genes, or we have 2 alleles for each character. So monohybrid individual produces 2 gametes. Dihybrid individual produces 4 gametes. Now the trihybrid produces 8 gametes. So the number of gametes produced is increasing geometrically from 2 for monohybrid, 4 for dihybrid, and then 8 for trihybrid. Now what are the gametes produced? How to remember those gametes? What are the gametes produced by this F1 trihybrid? Now as we know, the number of gametes formed is equal to 2 power n. N is equal to the number of heterozygous loci. Now we have 3 heterozygous loci. This is one, this is another one, this is another one. So we have 2 power 3, 8 possible gametes are produced. 2 power 3, 8 possible gametes are produced. Then how to remember the gametes? We have to take the first 2 letters 4 times each. 4 times each. The next 2 letters 2 times each alternate. And the next 2 letters just only 1 time. So this is the arrangement. Taking the first 2 letters, write each letter 4 times. Then taking the next 2 letters twice each. Capital R, then small r, small r. Then the next 2 letters alternate. I am taking for example the first one, taking just the first letter 4 times. And taking the second letter 4 times. So this is over. The first 2 letters, each one I have written 4 times. Then how to take the next 2 letters 2 times each. For example, here I am writing capital R, here writing capital R. Then writing small r. Here also the same way, capital R twice, small r twice. Now this is about. Now we have to take the third pair of letters. Write the letters alternatively 1 time each. For example, capital Y, small y, capital Y, small y. Here also capital Y, small y, capital Y, small y. So these are the 8 gametes produced. These are the 8 gametes produced. This is the way of remembering the gametes. Taking the first 2 letters, writing 4 times each. Taking the next 2 letters twice each, just for the dominant letters and also for the recessive letters. Now we have received 8 gametes. Now the probability. Each each gamete, so this is suppose the male gamete. Similarly, 8 gametes are produced by means of, that is the female individual. All these gametes are formed because of independent assortment of the genes. Now on either side we have 8 gametes. Each gamete has equal probability of receiving actually one of the 8 gametes from each pair. So in a zygote, we are getting actually a combination of both male and female gametes. Now the probabilities are 8 into 8. So totally 64 individuals are produced. The number of gametes produced is equal to 8 and the number of individuals produced, the probability 64. I mentioned already, the phenotypic ratio normally equal to the number of gametes produced by a trihybrid. In monohybrid 2 gametes, so we are having the ratio 3:1. Now in the case of dihybrid there are 4 gametes, so we are getting 4 phenotypes, 9:3:3:1. And similarly here, the number of gametes is equal to 8 and so also the number of phenotypes, the F2 is equal to 8. Now we have the following proportion. What is the phenotypic ratio obtained? We are talking about only the phenotypic ratio, not any other category in your syllabus, even for the entrance examination. Now the phenotypic ratio, I mentioned already, we have 8 phenotypes. The first one 27, the next 3 are 9, the next 3 each, and the last one 1. So this is the phenotypic ratio 27, 999, 333, of different combinations of 8 types of phenotypes. Now in the case of genotypic ratio, or the total number of genotypes, in monohybrid I mentioned we have, that is 3 genotypes, taking the first number, the largest one. In the dihybrid, for remembrance, the largest number 9, so 9 genotypes. And here the largest number in the phenotypic ratio is 27, so we are getting 27 genotypes. So the number of genotypes, for these 8 phenotypes 27. For monohybrid 3 genotypes, then dihybrid 9 genotypes, then for, that is trihybrid 27. So 9 into 3 we are getting, that is 27 for easy remembrance. So 3, 9 and 27. That's about the trihybrid cross. In your syllabus given only the ratio, what is trihybrid cross and how it can be produced, etc. the definition. In addition to, that is monohybrid, dihybrid and trihybrid crosses of Mendelian inheritance, there are also certain exceptions. That is nothing but the occurrence of different phenotypic ratios. We have different phenotypic ratios, the modified ratios when compared to 9:3:3:1. So what is that one? We have the different patterns of inheritance. The different patterns of inheritance normally resulted due to different phenotypic ratios. We have examples. There are examples in which the phenotype of an organism is a result of the interaction of 2 or more genes. So that is what we have exceptions to Mendelian inheritance. So the complex pattern of inheritance, the complex patterns of inheritance are nothing but the exceptions and also the extensions of Mendelian genetics, extension of Mendelian genetics. Now we have under this one, what is gene interaction? What is gene interaction? Now each phenotype is controlled by more than 2 sets of genes. each phenotype is controlled by more than two sets of genes. And each gene has two or more alleles. And this is called gene interaction. Each phenotype is controlled by more than two sets of genes. And each gene has more than two alleles. Or each gene has two or more alleles. And this phenomenon is called gene interaction. So many interactions we have. So many phenotypic characters normally due to, for example, the many expressions, many interactions, including the phenotypes, which include the structural and biochemical, which constitute the phenotype of an organism, are controlled by the interaction of two or more genes. So many characteristics of an organism, including the structural as well as the chemical characters, which constitute the phenotype of an organism, are controlled by two or more genes. This is what we call gene interaction. Now, under this one, we have to know what is factor hypothesis. What is factor hypothesis? As we know, according to the Mendelian inheritance, each character is controlled by a single gene. But we have exceptions. There are different exceptions with the different just interactions occur between the genes. In addition to that one, so we have different exceptions or various exceptions in which there are different interactions between the genes. This is a possibility, and this is called factor hypothesis. The occurrence of variations, the occurrence of, you see that one, genic interactions that result in two or more genes. That is called factor hypothesis. So genic interaction occurs between the characters. That is called factor hypothesis. The concept of factor hypothesis was proposed by Bateson. The concept of factor hypothesis was proposed by Bateson in 1905. And hence it is also called Bateson factor hypothesis. Bateson factor hypothesis. So the occurrence of, that is, different phenotypic ratios. This is because of the interactions between the genes, and that is called the factor hypothesis. As per the Mendelian concept, one character, one gene. Here different genes are interacting together to produce one phenotype, and that is called Bateson factor hypothesis. According to Bateson factor hypothesis, there are two types of gene interactions. What are the two types of gene interactions? One, intragenic interactions. Intragenic interactions. It's also called intralocus interactions or intra-allelic interactions. Intra-allelic interaction, or simply we can say allelic interactions. Allelic interactions. Now interactions occur. In the case of intragenic interactions, interactions occurs normally between the alleles of the same gene. For example, if you are taking a gene A, so interactions occurs between capital A and small a. So these two are located in the same locus. I am taking locus 5. Here we have the gene A, small letter recessive. So if interactions occur between the alleles of the same gene, then it's called intragenic interactions. We'll talk about later. So it is intralocus means occurring at the same locus, or intra-allelic, capital A and small a are alleles. As simply we have allelic interactions. Another one, what we have the second type, interallelic interactions. The second type, inter. That is in between. Intra means within. Inter means in between. Intergenic interactions or interlocus. I am changing the word. Interlocus interactions or interallelic interactions or non-allelic interactions. Intergenic interactions or interlocus interactions. Interallelic interactions or non-allelic interactions. Here the interactions occur between two different genes, for example A and B. These two are non-allelic genes. So capital A, small a are allelic genes. These two are alternative forms of the same gene. But capital A and capital B are non-allelic genes located in different loci. That's why it's called interlocus, between different locus. And that one is called intergenic interactions or interlocus interactions or interallelic interactions. Okay, so that's about the two types of interactions according to Bateson. Based on Bateson factor hypothesis, the genic interactions are classified into two types. Number one, intralocus or allelic interactions or intragenic interactions. So here the interactions occur between the alleles, capital A versus small a. That is called intragenic within. Another type, intralocus or non-allelic interactions. Here it occurs between two different alleles, capital A and capital B or small a, small b, like that. Now, the intralocus or allelic interactions are studied under three different headings. One, the dominance relationship. And this we have different types of genic interactions we are going to study soon. One, complete dominance we already seen. That is between tau and the dou, the appearance of one character in the F form. Then the next one, incomplete dominance. The third one, codominance. And the last one, overdominance. Now likewise the second type of intragenic interactions, the lethal genes. Now lethal means killing effect, and which we have the following types: dominant lethals, recessive lethals, conditioned lethals, sex-linked lethals and balanced lethals. These are all coming under the heading lethal genes. Now the last one, multiple alleles. This is also coming under intragenic interactions. The best example you know, you studied already, the blood group. Now coming back to interlocus or non-allelic interactions. Here there are two types, epistatic or hypostatic interactions. One gene suppresses the expression of another gene. We will talk about later. Epistatic or hypostatic interactions. The second type, additive. Polygenic inheritance. What is studied? Quantitative genetics. Quantitative genetics, here we are studying about the hereditary traits of a group of organisms where the genes, many, they control the character. That's called polygenic interactions. Additive interactions also another name. Also we can say multiple factor hypothesis, not multiple alleles, multiple factor hypothesis. More than two genes affect the expression of a character. So these are all the different types of major classifications. We have to go one by one, some examples, and the intralocus or allelic interactions, and then interlocus and non-allelic interactions. One example for epistatic interactions. Another one, polygenic interactions, in the case of kernel color, in the case of pattern. So this is about the general classification. We'll talk about later one by one, that is in each topic.